In this video, we will talk about the etiology and pathogenesis of multiple sclerosis. And we are going to start with the etiology, that means the causes that induce this disease. Multiple sclerosis has a complex etiology involving different factors, some of which have already been identified. One of these factors is the genetic background. Although multiple sclerosis is not a genetic disease, there are genes that increase our susceptibility to develop this disease, and most of these genes are associated with the immune system. These multiple sclerosis susceptibility genes are believed to have originated in Northern Europe, in what is now Iceland, Scandinavia, and the British Islands, which are in fact the countries where the highest prevalence of the disease is observed until today. And from there, the MS risk genes spread through Europe to Asia and North Africa with the Viking invasions, in what is known as the Viking Saga. As the invasion moved away from the starting point, the genetic component was diluted by the Vikings mating with women from the occupied areas, and in fact today the prevalence of MS in these countries is decreasing if we move farther away from the Nordic countries. Subsequently, there was another expansion of the genes towards what is today Canada, USA, Australia, and New Zealand due to the colonization of these territories by people from Northern Europe. The fact that this colonization was carried out mainly by families and there was little mixing with the autochthonous population has resulted in the prevalence of MS in these countries remaining similar to that of the areas of origin, that is high. The higher prevalence of MS in people genetically related to affected people supports the relevance of the genetic background in this disease. If a person is not related to a person affected with MS, the prevalence is approximately 1 in 1,000. If a person is a paternal half-brother or sister, the prevalence increases to 13 in 1,000. If a person is a maternal half-brother or sister, the prevalence increases to 24 in 1,000. In other words, the risk is greater if the mother is shared than the father. And this is because it is believed that genetic components that are present in the mitochondria, which we only receive from the mother, could influence susceptibility to multiple sclerosis. Mitochondria are a part of our cells, and they produce energy and have their own DNA. If a person is the brother or sister of a person with MS, the prevalence increases to 35 in 1,000. If, in addition to being a brother or sister, they have an identical set of HLA genes, that is the histocompatibility complex, the prevalence increases to 80 in 1,000, and in genetically identical twins to 150 out of 1,000. Although these data clearly support the relevance of genes for this disease, the fact that the concordance in genetically identical twins, that means that both suffer from MS, is not 100%, suggests that there must be other factors that play a role in its development. And these factors, if they are not genetic, have to be environmental. As we just commented, the distribution of MS in the world shows a higher prevalence in Northern Europe, Northern America, Australia, and New Zealand. This distribution reveals a latitude gradient according to which the prevalence increases as we move away from the equator. As we have also seen a moment ago, migrations are probably the main factor that is responsible for this distribution. However, this latitude gradient is also observed within countries such as the United States or Australia, where there are no major genetic differences between the areas closest to and furthest from the equator that can explain it. So, the existence of this gradient suggests the involvement of environmental factors in the development of multiple sclerosis. Studies in migrant populations also support the involvement of environmental factors in the development of MS. In studies of people who have migrated from high-prevalence areas such as the United Kingdom to low-prevalence areas such as South Africa, it has been seen that if these people migrated before the age of 15, they acquired the low risk of suffering MS from the host country, while if they migrated after the age of 15, they maintained the high risk of suffering MS from their country of origin. In studies of people who have migrated from low-prevalence areas such as India to high-prevalence areas such as the United Kingdom, the results obtained were very similar. Children of Indian immigrants born in the UK are at higher risk of developing multiple sclerosis than people born and raised in India. Therefore, all these data indicate the existence of environmental factors that can influence the risk of developing multiple sclerosis. Among the factors identified so far are some viral infections, such as the Epstein-Barr virus, which is a herpes virus that can induce infectious mononucleosis, a disease also known as the kissing disease, sun exposure, and therefore vitamin D levels, and some habits such as smoking and also the diet, since obesity during childhood and adolescence increases the risk of MS, and the gut microbiota can play a very important role in this disease. The relevance of each of these factors may vary from one person affected by MS to another. While genetics may be the most important factor in some people, infections may be the main trigger in others. The etiology of multiple sclerosis is therefore very complex, like its pathogenesis. Different mechanisms are involved in the pathogenesis of MS, in particular, inflammation, demyelination and remyelination, oxidative stress, astrogliosis, metabolic disturbances, and neurodegeneration. In the central nervous system, the axons of neurons are covered by myelin produced by cells called oligodendrocytes. Other cells, called astrocytes, are important for supporting neurons. After autoreactive T lymphocytes have been activated in the central nervous system, inflammation is induced. And as a consequence of this inflammation, demyelination and oxidative stress occur. Although there is some capacity for remyelination, it is limited, and if inflammation persists, demyelination also progresses. As a consequence of demyelination, astrocytes are activated and accumulate in a process called astrogliosis. Furthermore, metabolic alterations are induced. After losing myelin, neurons die and neurodegeneration occurs. The relevance of each of these mechanisms may vary from one person with MS to another. Inflammation may be more pathogenic in some people than in others, while the remyelination capacity or the susceptibility of neurons to die after the loss of myelin may also differ from one person to another. The pathogenesis of MS is therefore very complex. Such a complex etiology and pathogenesis result in wide heterogeneity, that is, differences in MS between individuals. This heterogeneity affects the clinical course of MS with very different clinical forms, such as the relapsing remitting form, the secondary progressive form, the progressive form with relapses, or the primary progressive form. There is also heterogeneity in patients regarding the response to treatments, or in the distribution and characteristics of magnetic resonance imaging MRI lesions that reflect inflammation and neurodegeneration. This heterogeneity, which is still poorly understood, represents a major problem in the clinical management of people with MS. As we have previously mentioned, nutrition may act as a trigger for MS, but it can also influence the pathogenesis of this disease, since there are foods with nutrients that can influence inflammation and nutrients with neuroprotective and even remyelinating properties. In the next videos of this course, we will see how nutrients and diets can help us in the management of MS.